Hello friends, welcome to Ashu's Life Sciences. Proteins are incredibly complex structures, and their shape is critical to their function. Understanding the structure of proteins is essential for many areas of science, from medicine to biotechnology. Protein molecule. Proteins can have 4 levels of structure: primary, secondary, tertiary, and quaternary. The primary structure of a protein is simply the sequence of amino acids in the protein chain. The secondary structure of a protein is formed by hydrogen bonds between the amino acids. This can result in structures like alpha helices or beta sheets. The tertiary structure of a protein is the overall 3D shape of the protein. This shape is determined by interactions between the amino acids, like hydrophobic interactions and disulfide bonds. The quaternary structure of a protein is formed when multiple protein chains come together to form a larger complex. These chains can be identical or different, and the resulting structure can be very complex. The structure of a protein is critical to its function. Proteins can bind to other molecules, catalyze chemical reactions, and even act as molecular machines. Understanding the structure of proteins is essential for many areas of science, from drug discovery to biotechnology. By studying the structure of proteins, we can design new drugs that target specific proteins, or engineer proteins with new functions. In recent years, new technologies like X-ray crystallography and cryo-electron microscopy have revolutionized our ability to study protein structures. These techniques allow us to visualize proteins in incredible detail, revealing their shape and function in ways that were once impossible. Protein structures are incredibly complex and diverse, but by understanding their shape and function, we can unlock their incredible potential for improving the world around us. Proteins are incredibly important molecules that perform a wide range of functions in our bodies. Understanding their structure is critical to developing new treatments for diseases and improving our quality of life. The structure of a protein determines its function. By studying the structure of proteins, we can design new drugs that target specific proteins, or engineer proteins with new functions. Protein engineering is a rapidly growing field that is using our understanding of protein structure to create new drugs and therapies. For example, we can use genetic engineering to modify proteins to be more effective at treating diseases like cancer or autoimmune disorders. Proteins are also incredibly useful in biotechnology. They can be used as molecular machines to carry out specific tasks, like breaking down waste or producing energy. By understanding the structure of proteins, we can engineer new proteins that have specific functions. For example, we can design proteins that break down environmental pollutants or produce biofuels. In addition to their applications in medicine and biotechnology, proteins are also important for understanding evolution. By studying the structure of proteins across different species, we can learn about the evolution of life on Earth. We can use protein structure to create evolutionary trees that show how different species are related. This information can help us understand how different organisms evolved and how they are related to one another. Closing shot of the laboratory. In summary, protein structure is critical for understanding the function of proteins and for developing new treatments for diseases. It also has applications in biotechnology and can help us understand the evolution of life on Earth. By continuing to study protein structure, we can unlock even more of their incredible potential for improving our world. Protein structure details. Protein structure refers to the way in which a protein is organized and arranged in 3-dimensional space. Proteins are composed of long chains of amino acids that are linked together by peptide bonds. These chains can fold and coil into complex shapes, which determine the protein's function. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. Primary structure. The primary structure of a protein refers to the linear sequence of amino acids in the protein chain. This sequence is determined by the genetic code and can vary widely between different proteins. The primary structure is critical because it determines the protein's overall shape and function. Secondary structure. The secondary structure of a protein refers to the local folding patterns that occur within the protein chain. These patterns are stabilized by hydrogen bonds between amino acids. The two most common types of secondary structure are alpha helices and beta sheets. Alpha helices are coiled structures that resemble a spring, while beta sheets are flat and resemble a pleated sheet. Tertiary structure. The tertiary structure of a protein refers to the overall three-dimensional shape of the protein. This shape is determined by interactions between amino acids that are far apart in the primary sequence. These interactions can include hydrogen bonds, disulfide bonds, and hydrophobic interactions. The tertiary structure is critical because it determines the protein's function. Quaternary structure. The quaternary structure of a protein refers to the way in which multiple protein chains come together to form a larger complex. This structure is stabilized by a variety of interactions between the protein chains, including hydrogen bonds, disulfide bonds, and hydrophobic interactions. The quaternary structure is critical because it determines the function of the protein complex. The structure of a protein is critical to its function. Proteins can bind to other molecules, catalyze chemical reactions, and even act as molecular machines. Understanding the structure of proteins is essential for many areas of science, from drug discovery to biotechnology. By studying the structure of proteins, we can design new drugs that target specific proteins, or engineer proteins with new functions.